All right, I have 1 minute after 4, so why don't we get started? Good afternoon, everybody. Welcome to the Texas Epidemic Public Health Institute, aka TEPHI, Grand Rounds, and we're delighted to have with us this month Dr. Robert Atmar from Baylor College of Medicine. He is the John S. Dunn Research Foundation Clinical Professor in Infectious Diseases in the Departments of Medicine and Molecular Virology and Microbiology. He's a member of Baylor's Vaccine Treatment and Evaluation Unit and Digestive Diseases Center. And he also serves as chief of the Infectious Diseases Service at Ben Taub General Hospital. So you can tell he's a busy guy. We're delighted he was able to do this. And he's going to be talking to us today about noroviruses. But before I forget, I want to put in a little plug for our next Grand Rounds, which will be on September 13th, 4 o'clock Central Daylight Time, talking about GIS and public health. So, Dr. Atmar.
Thank you, Kathy. So I'm going to talk to you over the course of the next hour or so about human noroviruses. My title is Cruising Towards Prevention. And I'm going to show you that this is not just the cruise ship virus. So disclosures of interest in the past couple of years, there was a vaccine being developed by Takeda Vaccines, and we received research support from them. Hillevax took over the development of that vaccine, and I'm a consultant for them, and I will mention the vaccine towards the end of the talk. So the story for noroviruses dates back to October of 1968 with an outbreak of what at that time was called winter vomiting disease. The outbreak occurred in an elementary school in Norwalk, Ohio. Winter vomiting disease actually was first described in 1929 by a physician named Zahorsky. And there had been a number of studies trying to look at this disease up until 1968. This outbreak in Ohio was investigated by the CDC. There was a 50% attack rate. It was characterized by nausea, vomiting, diarrhea, and fever. The average incubation period was 48 hours. On average, the illness lasted for 24 hours. And importantly, there was a 32% secondary attack rate, which means that the individuals in the school took it home and gave it to their family members and other contacts. This samples taken from this outbreak were sent to the NIH, where Norwalk virus was subsequently identified by electron microscopy from persons who'd been challenged with the stool collected during this outbreak. And so fast forward, norovirus is the most common cause of gastroenteritis in the US, causing about 20 million cases on an annual basis based on estimates from the CDC. It causes both epidemic disease and endemic disease, affects persons of all ages, and can cause both symptomatic infection and subclinical infection. And I'll show you some examples of that in a little bit. The CDC has put together this pyramid, which shows at the bottom about the 20 million cases of total illnesses per year, resulting about 10% of those in outpatient visits, about 400,000 emergency department visits per year, 56 to 70,000 hospitalizations per year, and about up to 800 deaths per year. The 800 deaths are primarily in the elderly, and the risk overall for the population is about 1 in 5,000 to 1 in 7,000. Hospitalization lifetime risk is about 1 in 50 to 1 in 70, about a 1 in 9 chance of going to the emergency room at some point in one's life, a 50-50 chance of having a norovirus infection that leads to an outpatient visit, and at least 5 lifetime illnesses on average in the US. This is a virus that is genetically and antigenically very diverse. There are currently 10 genogroups that are recognized, with another couple that have not yet been named, because there's only one member in each. The genogroup 2 viruses are by far and away the most common, causing up to 90% of the human infections that are recognized. Genogroup 1 is the second large genogroup, with Norwalk virus being the prototype G1.1 strain. And this causes anywhere from 10 up to 20% of outbreaks and human infections. And then there are some other uncommon strains in the genogroup 4, 6, or 4, 8, and 9 in terms of causing human disease, and a bunch of other animal genogroups that do not cause infection in people. The G2 viruses are further, and the G1 viruses are further subdivided into genotypes, with G2.4 being the most common, causing anywhere from 50 to 80% of infections. And this genotype, the G2.4, undergoes antigenic drift, similar to what we see with influenza viruses. Between 1980, the late 80s and early 90s, about every 3 to 5 years, a new variant emerged, up until 2012, which is not shown on this slide, but no new variants have emerged since then. And this emergence of new variants has been thought to been driven by immunity within the population, although we don't currently know why after 2012 no new variants have emerged. So that's one of the unanswered questions. But this does pose a potential challenge then for developing preventive strategies that are based on vaccination. Outbreaks of human norovirus disease occur almost anywhere that people gather, and many of us who were around in 2005 recognized that there was a hurricane, Katrina caused a major catastrophe in New Orleans, in Louisiana, and a lot of the evacuees from New Orleans came to Houston, and many were put up in the Astrodome complex. And the picture in the lower right shows how these people were boarded. Well, early on after they arrived here, starting on September 2nd, a gastroenteritis infection was recognized, and essentially increased in numbers over the ensuing 2 weeks, and it really was not dispersed until all these people were dispersed into the community and no longer held as a group in the Astrodome complex. The overall attack rate was estimated to be 18% there, and it was a G217 virus that caused the outbreak. But essentially, any place people come together, we have the potential for norovirus outbreaks to occur. And we're going to talk a little bit more about what the most common settings are in a few minutes. The CDC tracks norovirus infection throughout the year, and the gray bar on this graph basically shows the average number of cases or expected number of cases over the course of the year. And so you can see that in the winter months, essentially from November up into March, is when noroviruses are most prevalent. Although they cause...
Robert, we're not seeing that. Yeah.
Sorry,
okay.
I forwarded the wrong slide. Sorry, right here.
There we go.
So the gray is the 95% confidence intervals of when norovirus is most likely to occur, and the numbers of cases averaged over a little bit more than a decade. And you can see that there's a winter seasonality, although illness occurs throughout the year. And the other thing that this figure shows is that COVID did have an effect on the seasonality, although in the current year, we seem to have recovered back to something that's more similar to what we were seeing pre-COVID. If you look at the settings of norovirus outbreaks, over half of the ones that are reported to the CDC occur in long-term care facilities, a little bit more than 10% in schools or daycares. Restaurants make up another 10% or so. And cruise ships, which aren't shown here, but fall in this other single settings, make up only about 1%. So even though we hear a lot about cruise ships being associated with norovirus outbreaks, they're really just a very small minority of all the outbreaks that occur. And if we look at how these outbreaks, the predominant means of transmission is predominantly person to person, with food being about 14%, water being less than 1%, and then other means largely unknown, making the most of the rest of the fraction. Now, I should also mention that many outbreaks might begin as a foodborne or waterborne outbreak, and then the means of transmission can be through environmental contamination or from person to person spread. So in other words, transmission in outbreaks can be mixed. The clinical manifestations are basically those of gastroenteritis, watery diarrhea being characteristic, but in outbreaks, or in norovirus in particular, vomiting is a very common symptom. And if you have an outbreak in which more than half the individuals involved with a diarrheal illness have vomiting, it makes it very likely that it's going to be a norovirus-associated outbreaks. There are other symptoms that are often present, and the main complication is volume depletion. You can have electrolyte abnormalities. And then in immunocompromised patients, instead of just acute gastroenteritis, they can have a chronic illness that can lead to severe malnutrition. In other parts of the world, malnutrition in young children can be a problem too. And then there's some rare complications that can lead to death. Now, for many years after the initial discovery or description of noroviruses in 1972 as being Norwalk virus as being a cause of gastroenteritis, the only way to study the virus was either in investigating outbreaks or doing controlled human infection model studies. And it was only in the last probably 7 to 8 years that we've been able to grow the virus in human intestinal enteroids as developed in Mary Estes' lab here at Baylor. Back in the 2000s, we did a controlled human infection study where we enrolled adults who were 18 to 50 years of age and gave them either placebo or live virus. And the purpose of this study was to try and determine the human infectious dose. Our volunteers, study participants, were screened for eligibility. They were admitted to the hospital on day 0 and inoculated with different doses of virus. They stayed for a minimum of 4 days or 96 hours and were discharged only after they'd recovered for at least 18 hours from their symptoms. And then we got samples at different time points after discharge. And in terms of trying to answer the question about the human infectious dose 50, we use different doses of virus that are shown, the doses, inoculum dose in genomic copies, log 10, shown across the x-axis, and the percent infected is shown on the y-axis. And what we saw is that at the higher doses of virus, of Norwalk virus that we used, we essentially infected 100% of the people who were genetically susceptible to it. And then gradually fewer people with a HID 50 estimated to be just over 1,000 genomic equivalents. At the same time, there were investigators at the University of North Carolina that had their own challenge pool of Norwalk virus. And while they had a little bit more variability, the logistic regressions of the two studies came out to have very similar HID50s. Now, if you read the literature, the UNC group has estimated the ID50 may be as low as 18 genomic equivalents, 18 virions, where you can see in their study, they didn't infect anybody until they got up over 1,000. And they did this, I think, by some hand waving, but essentially they made the assumption that infectious virus could be clustered together. And if they took the clustering clumping into account, they got this much lower estimate. But I'm showing my bias that I think, you know, the HID50 is around 1,000 for Norwalk virus, and in cell culture we see up to 10 to the 4th TCID50 or genomic equivalents to make one TCID50. So somewhere in here. If you look at virus shedding from some of the volunteers who were in this study, these are GE stands for gastroenteritis in this slide. This black rectangle here is a period of time when people were symptomatic, so between 1 and 3 days after challenge. And we took all the stool samples generated by, you know, these 5 different volunteers, and you can see that they shed a lot of virus on the y-axis, up to 10 to the 11th copies or genomic equivalents per gram of stool. And for some, the peak virus shedding was actually in the post-symptomatic period. And the other thing is that these people, even after recovery, shed high levels of virus out for a week, and even measurable levels out to a couple of weeks. And we found very low levels out to as late as 6 weeks. What we don't know is whether all of this virus is infectious or not. But if you look at the amount of virus being shed from infected persons, there are up to 1 million HID50s of virus present in 1 microliter of feces at the time of peak virus shedding. Up to 1,000 HID present in 1 microliter of feces for 7 to 10 days after illness onset. And so recognize that this is 3 to 5 days after recovery from symptomatic illness, and up to 10,000 HID present in 1 ml of vomitus. So 1,000 times larger volume, but still a lot of virus present in emesis. So now I'm going to switch to talking about susceptibility. And I did mention this idea of genetic susceptibility. And there, from some of the, both the outbreak studies and these challenge studies, we found that there, for some of the norovirus genotypes, there is a genetic factor that makes people, if they don't have it, resistant to infection with Norwalk virus and with G2/4 viruses. Saying that another way, people are secretor positive. They are genetically susceptible to some genotypes like G11 and G2/4. So what is the secretor status? This is essentially determined by the presence of a fucosyl-2 transferase gene. And this gene modifies glycans that are expressed on the surface of epithelial cells within the gut to express the ABH glycans, or the blood group antigens. And so genetically, people who have this functional fucosyl transferase 2 gene are genetically susceptible to G11 and G2/4 viruses. And those who are secretor negative and don't have a functional FUT2 enzyme are genetically resistant. That said, there are some genotypes that don't seem to have this secretor restriction. And so essentially all people can be infected with some genotype of noroviruses. But the G24 are the epidemiologically most important, as I mentioned before. And I'll show you a little bit more about that in the next slide. Now, the other thing for susceptibility is that we know that there's adaptive immunity that occurs in that there can be short-term resistance, and short-term means from months to a year or two, resistance to reinfection and illness. And we correlate that, at least in part, with serum antibody levels or mucosal IgA levels directed specifically against a virus strain. Similar to what we've seen with other viruses that are transmitted and cause disease at the mucosal surface, like influenza. This is a nice graphic that just shows the secretor phenotype with the darker shades having a higher percentage of the population. So the darkest shade here, like in Mexico, 81 to 94% of the population are secretor positive. The next darkest is 78 to 81%, as in South America. Some of the countries in South America being secretor positive. In the US, 76 to 78%. And this lightest pink is 49 to 71% parts of Asia and South Africa. Accompanying this slide in this paper a couple of years ago in JID was a similar slide or graphic showing G24 frequency in terms of causing outbreaks and disease in these countries. And it largely mirrored the secretor phenotype distribution. So virus transmission, we know that relatively low infectious dose, 18 virions, 1,000 virions, still a relatively low dose that can be transmitted and cause disease by very small volumes of feces contaminating an environment, food, or whatever. These viruses are relatively resistant to inactivation, and I'll show you that a little bit more when we get to talking about ways to try and disinfect or inactivate the virus. The predominant route of transmission is fecal-oral, food, water, person-to-person being the major cause in outbreaks, or environmental contamination, either through fomites or by just contamination of the environment after a vomiting event. And then there's some evidence that it can occasionally be airborne. I, in particular, like this report that showed up a couple of decades ago, where this index case was at this table and had vomited. And then these other blocks represent the other tables. And this larger rectangle represents tables in an adjacent room that had a connecting doorway. And the further you got from ground zero, if you will, where the index case was, the lower the attack rate was. But there were even people in this adjacent room who became ill and had no direct contact either with the index case or the environment around this table. So this suggests that the virus may have been spread from the vomiting episode in a small particle aerosol into the adjacent area. And that the closer you got, maybe the higher the inoculum dose. There have been other outbreaks in emergency centers where people just walking through an emergency center became infected, suggesting airborne transmission. But this is not the major way of transmission and would be a minor way at best. Now, the other thing is that we've seen outbreaks occur pre-symptomatic. So in that challenge study, we found virus in the stool up to 3 to 6 hours before persons became symptomatic. During symptoms is the highest risk of transmission, but post-symptomatic and even asymptomatic or subclinical people have been able to transmit it. This study out of the Netherlands showed that in looking at nosocomial outbreaks of norovirus disease, that the symptomatic patient transmitted the disease most efficiently. Symptomatic staff with an R0 or R effective transmission still greater than 1. Asymptomatic patients occasionally transmitted, and they didn't recognize any from asymptomatic staff, but that potentially could still occur. So it really is during the symptomatic phase, and one might say that could be either because that's when the highest amount of virus is present, but also difficulty in controlling both stool potentially and emesis that could lead to contamination of the environment and or the individual and transmitting it to person. The virus can survive in the environment. A study done at Emory where they used a relatively high dose. They took 10 to the 8th genome and put it into water samples and stored it at room temperature for up to 61 days. And then at various times during that 2-month period, took it out and gave it to study volunteers, and were able to infect people out to 61 days. So even at room temperature in the dark, the virus could survive for a long period of time. There have been outbreaks associated with norovirus RNA persisting in the carpet up to 72 hours after cleanup with detergent and water, where people became infected. And then norovirus present in shellfish implicated in outbreaks up to 2 weeks after a flooding event. So all of these data suggest the viruses can survive in the environment, resist either decontamination or cleanup or inactivation in the environment. Now, in terms of diagnosis, this can be made based on clinical grounds, and particularly in outbreaks. I mentioned the greater than 50% with vomiting as a symptom. If you're working up an outbreak and the incubation period is 1 to 2 days and the mean illness duration is half a day up to 2.5, 3 days, all of that suggests that the illness was due to norovirus. Now, this Kaplan came up with these criteria in the early 80s, where a lot of the focus was ruling out other bacterial parasitic causes. And Tercios reanalyzed data from the CDC in 2006 and found this definition, the Kaplan criteria, to have high specificity and reasonable sensitivity. That said, molecular diagnostics are the gold standard. There are some antigen detection assays that are commercially available, but they're much less sensitive, not much better than the Tercios reanalysis of the Kaplan criteria. And so molecular diagnostics are increasingly available in healthcare settings and in the public health departments, and is a major way to make the diagnosis. Now, coming back to some of the viral inactivation studies that were done through controlled human infection studies, the virus is acid stable, incubated at an acid pH like one might encounter in the stomach for up to 3 hours. 2 out of 3 people became ill after the virus was exposed to this acid pH. Heating it up to 60 degrees for 30 minutes, still a quarter of the people challenged became ill. And even with free chlorine at 5 milligrams per liter, over half the persons became infected. So this translates into using somewhere between 1 and 10% Clorox bleach solution to inactivate the virus. And then high hydrostatic pressure more recently has been shown to kill the virus, but the 60 megapascals can make certain foodstuffs like shellfish unpalatable. Personally, I think they're unpalatable anyway, but I understand many people enjoy eating raw shellfish. There've been, because the virus has been hard to grow, there've been a number of surrogates that have been used over the years. And so I've abbreviated them: Aichi virus, which is a picornavirus; feline calicivirus, which is really a respiratory calicivirus that infects cats; murine norovirus; Tulane virus, which is another calicivirus, but in a different genus, the ricoviruses; and then porcine enteric calicivirus, which is another calicivirus, but is a sapovirus. And the thing you can see looking across here, when 70% isopropanol or ethanol was used, that the amount of virus reduction shown on the y-axis really varies depending upon the surrogate that was used. And a lot of the EPA tests were based on using feline calicivirus. But murine norovirus, which is at least a norovirus, has very different characteristics. Of these, I actually think the Tulane best represents the human noroviruses. But more recently, we've been able to grow human noroviruses in cell culture, and these are data from the CDC, which basically showed that neither isopropanol nor ethanol completely inactivated human noroviruses, and you needed to use chlorine at greater than 800 parts per million to consistently inactivate the virus infectivity. These data were published in 2018. So treatment is really symptomatic. There are no licensed therapeutics, and so trying to provide symptom relief, analgesics for discomfort, antiemetics for the vomiting, and antimotility agents for the diarrhea, and then fluid replacement if volume depletion is a concern. There are a number of interventions that have been used anecdotally, and I have to say these are anecdotal, and there are many examples in the literature where these therapies have not been beneficial, primarily in immunocompromised individuals. There actually was a phase 2-3 clinical trial conducted by the NIH in immunocompromised transplant recipients that was stopped early in the COVID pandemic, but and it's not yet been published, but the results are available on clinicaltrials.gov, where it didn't have any effect on the clinical outcomes for the patients in that study, and it had no effect on the levels of virus being shed in the stool. Similarly, immunoglobulin ribavirin, there have been anecdotal cases where patients have improved, but whether it was related to the treatment or not is really unknown. So then that brings us to how do we prevent infection with noroviruses? And the main thing is interrupting transmission. So to do that, we have to recognize that there is an infection occurring. We need to isolate the ill individual and exclude that person from circumstances where they might be able to transmit it. And then we have to inactivate the virus primarily through disinfection means if the environment's been contaminated. There are attempts to develop a vaccine, which I'll tell you a little bit about. And then there are studies really more laboratory based at this point to try and develop antivirals. Really, there aren't any that are currently in clinical trials that show any promise to my way of thinking. So what about non-pharmaceutical interventions? We know during COVID, that this was a study that was published a couple years ago now in JID, that during the pandemic, the number of outbreaks significantly decreased compared to the pre-pandemic period. These are different states that were involved in CDC-related evaluation of outbreaks, and in a number of different settings. There were either significant or trended towards decreased risks of norovirus outbreaks. So, some of the non-specific measures that were taken to decrease the risk of COVID also translated into human noroviruses, much like we saw with flu and RSV in that first season. Exclusion is an important means of decreasing the risk of transmission to others. We try and do that in healthcare and in childcare, and in particular in the food industry. Although many in the food industry, it requires a specific diagnosis to require food handlers from returning to work. And usually in many states, they're only excluded for 24 hours, but the CDC actually recommends up to 48 to 72 hours after symptom resolution. And you can see why from the data I presented earlier in terms of when people were shedding large amounts of virus in their stool. Much beyond 72 hours just isn't practical, and even beyond 24 hours becomes problematic. And there are some places in the country where regulatory authority requires approval for people to return to work in the food industry. Isolation in a hospital setting or in a healthcare setting, contact precautions, preferably in a single room. If you have an outbreak going on, patients can be cohorted, and the duration, again, is recommended by the CDC to be at least 48 hours after symptom resolution. And longer periods might be considered in younger children because they shed for a longer period of time and at higher levels. And also the same is true for immunocompromised patients like those with hematologic malignancies or transplant. The hand hygiene that is preferred is using soap and water with hand washing for at least 20 seconds. And that's because the alcohol-based hand sanitizers don't disinfect or kill the virus, as I showed you in some of the few slides ago. Education, especially in persons returning from illness, about the importance of hand hygiene, and because they can still be shedding high levels of virus in their stool. So, paying attention to those signs as you leave the bathroom in the restaurant about washing one's hands, particularly the food handlers before returning to work, is really important. And then no bare hand contact with ready-to-eat foods in the food industry. So making sure your food handlers are wearing those gloves as they contact food. For environmental disinfection, pre-cleaning to decrease the organic load is really important before it's exposed to either further disinfectants or steps to inactivate the virus. A bleach solution is the most reliable way of killing the virus, although this can be problematic because of the effects that bleach have on equipment and particularly things like carpet. And the phenolic and quaternary ammoniums are less reliable disinfectants for the noroviruses. Additional measures, particularly in outbreaks. We don't recognize outbreaks in healthcare settings as much in the US as they've been reported in the UK and some European countries. But in settings where there are outbreaks, restricting patient movement and transfers, restricting visitation, and even closing a ward to new admissions are all measures that have been effective in the UK in helping control outbreaks. What about vaccines? We actually have something to talk about. There several different strategies that are being investigated. Virus-like particle vaccines that are expressed through different systems, baculovirus expression, yeast-based systems, plant-based systems as Medicago, recombinant adenovirus, and then mRNA-based. The mRNA-based CureVac initially has been doing some preclinical work. Moderna earlier this year noted that they're interested in developing a human norovirus vaccine. But the main two I'm going to focus on are Vaxart and Hillevax, and they're actually in phase 1 and 2 clinical trials, with Hillevax potentially starting a phase 2/3 trial later this year. So the Vaxart platform, they have a recombinant adenovirus type 5 vector that has an E1, E3 deletion, and it expresses the, they put the VP1 gene into this adenovirus vector to express the VP1 antigen. And it also has an immunostimulant by making double-stranded RNA that serves as an adjuvant to increase the immunogenicity of the vaccine. And they've used this platform for papillomavirus, for influenza, and for norovirus. And with norovirus, they've been able to show that they can get an antibody response to the immunizing strain up to just under 4-fold compared to placebo and compared to baseline antibody levels. This isn't very impressive compared to what I'll show you with Hillevax, but the thing with this vaccine is it's delivered inorally. It's given in adults as a capsule and replicates, has abortive replication in the gut to immunize directly the gut, and might work much like the nose drop vaccine for flu works with an attenuated, live attenuated influenza vaccine. They've not been able to do any proof of concept studies that, at least that they've reported out yet, to show that it does prevent infection and illness, but those studies are ongoing, and hopefully we'll hear more about that in the coming year. The Hillevax vaccine was previously being developed by Takeda, and before that by Ligocyte Pharmaceuticals. And Ligocyte initially licensed this from Mariestis and Baylor as a VLP-based vaccine. What they're currently developing is a bivalent vaccine containing a G1 strain and a G2 strain, the G2.4, and they've vaccinated over or enrolled more than 7,000 people in studies to date. The studies have shown that in adults, a single dose is sufficient to immunize adults, but children, they're giving more than one dose, particularly children who have not been primed previously. They had initially done some studies with MPL as an adjuvant added to alum, but found that wasn't necessary. And their immune responses to the G1.1 component have been more robust. So they're actually using more G2.4 in the vaccine compared to the G1.1 component. They're using 15 micrograms of the G1.1 and 50 micrograms of the G2.4. The G2.4 actually is a consensus strain made from 3 different G2.4 genotypes or variants, I'm sorry, that were described. Houston 2002 as a backbone, and then with some of the epitopes that have been described for G2.4 being changed to either 2006A or 2006B. This is all with alum as an adjuvant. And here you can see the titers that were achieved by serum IgA in individuals reaching reasonably high titers within a week titers peaking at a week and then declining over the ensuing year, with the response being higher to G11 than to G24, but all persisting at levels higher than were present at baseline. And what these, the black dots represent people that were 18 to 60 years of age, I think it was, and then the open square and triangle were older individuals, which show you that essentially the responses were similar regardless of age in these healthy adult studies. In terms of looking at potential efficacy, the initial study was a study that I helped lead, looking at intranasally delivered G11, where we showed protection from illness, significant reduction to show proof of concept that illness could be prevented. And then they did the same study with a G24 challenge strain, and the primary outcome, they did not achieve statistical significance, but they had a very low attack rate in the placebo group, only under 40%. But if you looked at moderate to severe or severe, they trended more closely to preventing that. And then they did a field trial in young, healthy Navy recruits, where they showed significant reduction in moderate to severe AGE from any cause. And importantly, most of these cases were caused by G22 virus, which is antigenically quite distinct from the G24 in the strain, showing that heterologous protection could be achieved. This slide then looks at serologic correlates in the LVO3. This was a G24 challenge. And what we saw was that serum antibody that blocked binding of the virus to this histoblood group antigens, these glycans I was talking about, was significantly associated with the higher levels of antibody were associated with lower risks of infection, illness, or vomiting or diarrhea. And the same thing with serum IgA. But in those who got vaccination, we didn't see the same correlate, raising the question of the serum antibody after vaccination predict protection from infection or illness. And I think that's still an unanswered question. So some other studies have shown that vaccination with the Hilivax vaccine does lead to salivary IgA in primed adults. It does lead to cellular responses that are associated with protection. These are memory B cell, virus-specific memory B cell responses. And it does lead to antibody responses to non-vaccine strain genotypes. But unanswered questions that remain is, what is the importance of priming in vaccine-induced heterotypic protection? One of the things we've seen from epidemiologic studies in kids is that with their initial infection, their antibody response tends to be genotype specific. In other words, they develop antibody against the genotype that they were infected with, but not against other genotypes. Whereas in adults or older children with infection, we see not only homotypic with the infecting genotype responses, but we see heterotypic or heterologous responses to other genotypes. We don't know whether that is dependent upon what people have been infected with in the past. That's another unanswered question to be evaluated in future studies. We also don't know what the duration of protection is. This likely will be addressed in field trials, but because we know people can become reinfected throughout their lifetime, it's likely that boosts will be needed. But the question is with what frequency. What will be the target populations? Looking at efficacy versus burden versus economics. I think Hilivax is initially targeting pediatric populations where the greatest burden is in terms of burden of illness, whereas Vaxart may be targeting the elderly where the greatest healthcare burden is in terms of risk of mortality and hospitalization. And we also are learning how these new cell culture models will help us in vaccine development. We've been looking at neutralization of the virus using these cell culture models, and found that even though the neutralization assays are more difficult to perform, they correlate pretty well with the HBGA blocking assays, and which the HBGA blocking assays may be a better assay to, or more easier assay to use instead of doing neutralization. And then, whether live attenuated strains might be able to be developed or are out there and can be used as vaccines is unanswered. So I've taken you through a whirlwind description of where we are currently with noroviruses, tried to make it more of a public health bent, but I'm certainly happy if there's time and opportunity to answer any questions.
That was fantastic. And if you have questions, go ahead and put them in the chat. I'm going to take the prerogative of asking a few questions first. And I gotta say, Robert, I wrote down a lot of questions and then you answered them all. It was a great talk. One thing I'm a little unclear about, though, is cross protection between the different strains, which I know you addressed a little bit about vaccine. Are we, you mentioned an average of 5 infections per lifetime. Is that because of waning immunity or different strains or both?
So I think we don't really know that 5 infections per lifetime has been based on modeling studies that the CDC has done, or investigators at the CDC and at Emory School of Public Health have done. And if you think about 20 million infections per year in the US, now how much of the 5 infections is based on not being exposed, and how much of it is immunity? I think there are a lot of different factors that may play a role there. But we know from some of the controlled human infection model studies that people who were challenged with Norwalk virus and then re-challenged 2 to 3 years later could be reinfected and became ill with that reinfection. Whether that's because they were challenged with a high dose of virus and it really overwhelmed their immune system is hard to know. Those studies were done back in the 70s and 80s. So I think it probably is going to be a combination of exposure and waning immunity. I think that heterotypic immunity is probably going to be less good than homotypic immunity. In other words, if you're infected with G24, you're less likely to be infected with G24 if you're exposed compared to if you were exposed to G11. But we do know from many of our antibody assays that even with the assays where, when we do binding assays like ELISA, we see a lot of cross reactivity between the genotypes, and that's probably because I didn't really describe the VP1, but when the virus is formed, it kind of has a shell and then has protruding domains. The protruding domains are the ones that are responsible for binding to the cell and that a lot of the antibodies directed against. The shell domain, there are also antibodies directed against it, but it's not as well exposed on the virus, except maybe during binding to the cell, but it's much more conserved. And so in ELISA assays, it gets exposed and you see cross-reactive antibody responses. For the protruding domain, it's much more genotype specific, and there's less cross-reactivity in terms of being able to identify monoclonal antibodies that recognize multiple genotypes, but we have been able to make monoclonal antibodies, for example, that do bind to multiple G2 strains or multiple G1 strains. So there are some shared epitopes, and so we're still trying to figure out, which of those end up being protective. So long-winded answer, but I think the short answer is you probably have better protection, longer protection against the same genotype one was originally infected with, and that's what has led to the evolution of the G2/4 viruses in particular, to try and escape that immunity.
And one other question: how important is zoonotic spread in terms of where people are getting it from? And along with that, so what's the reservoir if most people only have acute infections? And I know you showed, at least they can be, I don't know if spreading is the right word because we don't know, but you can find virus up to a couple weeks, sometimes longer. But is it just immunocompromised folks who have chronic infections that are the reservoir?
So the question of the reservoir has been one of, and the emergence of new variants has been a topic of great interest in the field, at least for the last decade. And I think the current thinking is, well, there was a study that was published a few years ago showing, using modeling studies that is the immunocompromised population, which was a popular hypothesis a decade ago, they just aren't enough of them, and there's not enough, even though there is evolution in an individual, it doesn't explain the emergence of new strains. So the emergence of new strains is thought to probably come from children more likely than adults. They shed higher titer for a longer period of time. They have less background immunity. But that's still a hypothesis. In terms of zoonotic reservoirs, there are, so no animal strains have been able to be found in people. And yet there are human strains that have been able to be found in animals. So it's been G2/4 strains have been found in pigs. They've been found in cattle. There are notobiotic pig and cattle models of infection and some disease using those models with G2/4. But it's not thought that animals play a significant role as a zoonotic source, and we really have not been able to find, despite a lot of people looking, any animal strains in people.
Okay, and there's a question in the chat from Celia Clay. Given environmental persistence, carpeting, and depending on cleaning capability and small aerosol spread in an environment such as a cruise ship, what is the model recommended for stopping transmission? And I want to add on to that. You talked about bleach. Is that 10% bleach or straight?
No, it's 5 to 10%.
Okay.
Bleach is generally what's recommended. It may be as little as 1%, but so high level are using. Well, in a cruise ship, it's tough because once you have it in the cruise ship, it is very difficult to control. They basically end up going through and wiping down much of the ship between, particularly if they've had outbreaks on sequential cruises. A lot of what I described have been recognized through workup or evaluation of different outbreaks and been able to find the virus, for example, in carpet in a hotel where multiple persons who occupied a room became sick over time, and they found it in the carpet. And so that's where that was the only place they found it. So that's where they thought it came from. But you know, the problem with using alcohol or some of the other standard types of disinfectant is that they just don't work as well. And so, but you know, bleach is problematic.
Yeah. All right. There's a question about the slides. The presentation has been recorded. It will be available a week or two. You will get an email since you all have registered, and you'll get an email with it. I want to thank Dr. Atmar again. Fascinating talk. And I got to say, from a personal standpoint, I thought I had been told one virion could cause disease. So I'm glad to hear it's more like 17, I think you said.
18.
Yeah, 18. All right. All right. Thank you very much, everybody, for attending. And we will see you in September for GIS in public health.
Thanks.